Thank you, Derek. And I would like to extend my appreciation for ISPE for inviting me to talk about Raman technology. It also gives me immense pleasure to talk about Raman technology, which has actually come from India. I am from India. I'm working from Mumbai. So, Sir C.V. Raman actually got Nobel Prize in the year 1930 for discovering Raman technology. Well, a little bit about my background. So, my major is in chemistry, and then I did my PhD in organic chemistry. I did a postdoc with Dow Chemicals, and then I moved on to work in Reliance Industries as a research scientist for about 7 years. Then I worked as Raman and IR application scientist in Metla Toledo, and then for the past 4 years, I'm with this company called Endress+Hauser, and I'm working as Raman application scientist. Now, coming to today's topic, which is integration of Raman-based pad into biopharma. So, believe me, my education is in chemistry, and I'll be today talking about bit of chemistry and biochemistry and biology. So, let's see how Raman helps us in understanding the processes that we work, which eventually goes from a lab-based application to process. We get all kinds of drugs and chemicals out of the processes that we work on, but how this pad technology helps in really understanding those processes and eventually helps in making decisions for those processes. So the topics that I'll be covering today is the basics of Raman spectroscopy, comparison with other spectroscopic techniques, because we not only deal with this Raman, we also have a lot of other spectroscopic techniques that I'm sure everybody comes in contact on a daily R&D or the Q&C activities. Then advantages in bioprocess industry. Then, of course, the chemometrics, because this is really the heart of understanding the information that we get from all these techniques, and some case studies that I'll be discussing with you. Now, what are we talking about when we say analysis? So here, analysis methods, we use different light wavelengths. So we all know about UV, Vis, or Raman, tunable diode laser absorption spectroscopy, and NDIR, which is either NIR or IR technology. Basically, all these technologies use light of different wavelengths to study chemicals. So I'm sure UV Vis, many of you have already used. FTIR, NIR are also some of these technologies which are regularly used in laboratories. But Raman spectroscopy basically uses UV, visible, or near infrared light to measure chemistry. How does it really work? So when we say that light interacts with matter, you can have three different concepts here: either refraction or absorption or scattering. So molecules, when they refract, we have refractive indices to measure the refractive index or understand the refraction produced by those molecules. Absorption, there are UV Vis technology, NIR, or like I said, the FTIR technology, which are all based on absorption. Raman spectroscopy is based on scattering. Let's see how this scattering actually works. So when the laser shines on a sample, then basically these molecules interact with light and it begins to vibrate, because Raman is a vibrational spectroscopy. So the molecules begin to scatter. So when these backscattered photons are, when they reach the filter, then some of these scattering molecules are of the same wavelength as the laser. So they're blocked by holographic notch filter. But some of them are like what we call as frequency shifted. So their wavelength is shifted, and 1 out of 10 to the power 18 photons actually change the wavelength and they reach the detector. So when it reaches the detector, it detects those backscattered light as the Raman spectrum, and it gives the molecular fingerprint of the sample. This is the basic principle of how Raman works. And some of these this information is actually very crucial to the process, to the very crucial to the reactions that we are working on. And these shifts are what are called as Raman shift, and that is what we study. So Rayleigh scattering is basically the laser light of the same wavelength. That is not what we're going to study. We will study the Raman scattered light, which eventually goes to the detector, and we get the spectrum through the detector. Now let's see the comparison with some of the other spectroscopic techniques. So we have NIR, FTIR, and Raman. So NIR and FTIR are absorption-based technology, while Raman is, let's say, scattering or emission spectroscopy. What we get in NIR are mostly overtones, but with both IR and Raman, we get the fundamental information, and there are very distinct peaks corresponding to different molecular bonds in a sample. In order to prepare or study the NIR, no sample preparation is required. For Raman also, no sample preparation is required. While for FTIR, sample preparation is required if you want to study how it goes, unless we are talking about the online IR. But the main difference in all this is that water is a big problem in both NIR and FTIR technology. While in case of Raman, water is not an issue because it has actually, because of the fact that we don't get predominant water peak in Raman is used in life science industry. That is the main reason why Raman forms an integral part of the pharmaceutical industry or the life science industry. If we see the Raman and the mid-IR or FTIR spectra, basically Raman is complementary to mid-IR. So basically, what the bands which are actually strong in mid-IR are weak in Raman, and vice versa. What are weak in mid-IR are going to be stronger in Raman. But like I said, what really differentiates is the fact that there will be a very strong peak of water in FTIR 1640. But in case of Raman, you don't get that kind of peak, because of which, if you have to do the sampling or measurements of the water-based samples using FTIR, then all other peaks are completely submerged and you will not see those peaks, while it's not the case with Raman. Now, if we try to see how Raman and near infrared spectroscopy or the spectra, they look like, as you can see, I've taken a very simple spectra of acetone, which, while in NIR you have overtones, which we call as the big peaks, it's rather humps, but in Raman the peaks are very sharp. So you can first of all, when you just look at the spectra, you can see that the peaks are very sharp and it's very distinguishable, and it's very simple to read. So the quantification using Raman becomes much more simple as compared to NIR, and therefore in NIR you have to depend on the chemometrics to understand the progress of the chemistry. I'm not saying it's not required in Raman, but NIR compulsorily you have to see, because there are no peaks that you see in NIR. So basically, what advantages we see of Raman in lab and process environment? So Raman provides a molecular fingerprint with the specificity of infrared spectroscopy and the measurement ease of NIR spectroscopy. So just to give you an example of how it looks, in the fermentation process, as you can see, the Raman spectra of the fermentation process, the peaks are very, very sharp, even though 75 to 80% of it is just water. Sharp, the peaks are yet very sharp. Distinctly, you can see the glucose peaks or ethanol peaks. But the NIR spectra of the same fermentation process looks like this. So because of this major difference in how the spectra of both Raman and NIR look, Raman finds its advantages and its major use in the pharmaceutical industry. Now, when I say pharmaceutical industry, bioprocessing, so some of the advantages that you can clearly see, for example, you have the Raman and the NIR spectra of glucose and lactate in water. While we have very clear peaks of glucose, which is dark blue, we have lactic acid peak, which is green, and water, which is red. But as you can see, despite the presence of water, we have very clear and beautiful peaks there, while in NIR, it's just the same. So, unless you use algorithms, advanced mathematical techniques, you will not be able to study this kind of spectra. So, the main thing, fundamental thing that comes as a difference between Raman and NIR is that it's insensitive to water. Then you can have one probe for multiple measurements. So with the same probe, if you now focus on glucose or lactate, in this case, let's say water, so we are able to see all of them, just one probe, and we can have multiple measurements here. It measures the key process and quality parameters, and it's versatile to many applications. What do I mean by versatile to many applications? Raman finds its use not just in bioprocessing. We have Raman application in chemicals. We have these applications in oil and gas also. Believe me, many refineries are actually using Raman to study different processes in refineries, for example, gasoline blending and um, the hydrogenation processes in chemistry. So Raman finds its use in across industries. Then it is scalable from lab to pilot to the CGMP environment. So we can have the same unit or the same technology used in the lab. It can be extended to the process scale as well. Of course, it's inline installation, which means there is no risk of contamination and it's very low maintenance. So, because of these advantages, Raman has been accepted as a process analytical technology. It's a PAT tool. So, what do I mean by PAT tool? It is used in these processes, which are specialized systems for designing and analyzing and controlling manufacturing through timely measurement of critical quality and performance attributes. So it is not just a PAT tool, it is also involved in QBD, which is basically to design and develop processes that can consistently ensure a predefined quality of the product. So this actually forms a very important basis for how we look at the processes now versus what it used to be a few years back, and how the technology has evolved to incorporate all the changing environments of a process. So, process and quality measurements today versus future. So, today, what we do basically, we have to take a sample in order to understand what's happening in a lab or a process uh, chemistry. So suppose we have to take a sample, deliver it to the lab. We have to run the offline tests, wait for those results, and then of course validate those results. And then only we are in a position to say whether that batch is approved, whether the product that we're getting is of right quality or not. But in future, we can have this probe which can do inline multi-parameter measurement, continuously automated with AI and machine learning. We can also do the predictive analysis. So quality is the process and process is the quality. So right now, the as I said, Raman is a PAT tool. So we can do the real time analysis, real time release testing, and we can not only just do monitoring, process control, and quality predictions with this kind of online tools. So we are moved from real time analysis, moved from batch analysis, which used to be taking the samples. We are in a position to actually move to real time analysis with the probe installed in the reactor with continuous measurement. And like I said, it also helps in advanced process quality control, and it enables quality by design. So if you just look at how earlier we used to deal with the traditional bioreactors, we used to have the probe to measure DO, pH, temperature, the critical parameters, and then extracting sample offline measurements, and then decide the fate of the batch. But with inline Raman probes, you can have all this with proper optimization, quality assurance, with in a much reduced time. And this also enables the companies to to release their drug in the market at a much faster pace with enhanced product quality. So bioprocessing monitor and control targets. This I have highlighted this slide to tell you the kind of parameters that are studied in cell culture applications, in fermentation applications, and downstream purification. And with one probe, with one instrument, you can actually measure multiple process parameters. So for example, in cell culture, normally what do you measure? You have the nutrients like the glucose, glutamine. You can also measure the glutamate, lactate, ammonium concentration. Some of them are direct measurements because they are the molecules for which you have the molecular fingerprint on the spectra that you can study, but some of them are through indirect measurements like osmolality, viability, and even that we can measure using Raman. Similarly, in case of fermentation, we have glucose and sugar alcohols, all these, and many other metabolites. The picture that you see here is the spectrum, and I've highlighted the regions where you get to see the parameters that I've mentioned. So these regions have been studied, and looking at just one spectra with multiple overlapped with multiple spectra, you can actually get to the whole lot of parameters with one measurement. So Raman is very well suited for wide variety of in situ bioprocess measurements. One of the main reasons is that it's insensitive to water, because as you know, all these processes are mostly done in water. So now I'd like to talk about chemometrics. So basically, what is chemometrics? It is the study of process, which are the chemical based or the biochemistry based molecules using different tools. So when I talk about the process spectra here, all the spectra have been overlapped. And believe me, as a layman or somebody who is not seeing the spectra, they'll say, what is this? So many peaks just overlap. What am I going to get from this? Once you apply the chemometric tools to it, basically I'm talking about software which will look at specific peaks, which will work around those peaks and build algorithm, then this is the kind of uh, progress of the reaction or the process that people who are working on it would like to see. So the spectra overlaid here actually gives this kind of information. Now, what is this information? For example, in a chemical process, has my reaction started? In my reactant, is it getting consumed during the process? Is my product getting formed? Am I getting some impurities in the process? So that kind of information you will be able to get with just the spectrum. Okay, and how are we going to get it? This is only by chemometrics. Chemometrics is nothing but modeling. So there are a lot of software in the market where you provide the spectra, you provide offline measurements, there is a correlation, and you come up with the model. So the steps involved in quantitative analysis is first data collection. So which data I'm talking about? The two kinds of data. One, the data that we get from Raman. So it's a Raman spectra acquisition. So then once the spectra is acquired, we need the offline data from either GC or HPLC or any other analytical tool that you may be working on. So once we have the offline data and the Raman spectra, which is basically part of data collection, then we identify individual components of a chemical, biochemical, or pharmaceutical process. And then we do the sample testing to understand what are the distinct peaks. So once we have offline and the Raman spectra with us, we do a correlation, then we prepare a model. It's then calibrated, it's validated with some test samples, and a model is ready to predict. So we have three steps here: data collection, interactive model development, and automation. So once the first two steps are aligned, once you have the good spectra, which are correlating very well with the offline data you've been given with, then the model that we prepare, like I said, different algorithms and different software, that model should be able to predict what is happening in your process. So, for example, this is showing some critical process parameters which are generally used during cell culture process. So I have just taken examples of glucose, lactate, the enzyme viability and all. So the black line that you see is a Raman spectra, and the white dots are the offline data given by the the customer or the people who are working on it. So as you can see, Raman spectra is indicating how the process is going. Excuse me. So offline data tells you what happened, and online data tells you what is happening and what may happen, and you can take control of the whole situation of the process. I'm sorry. Okay, now let's look at some of the case studies here. So, with the abundance of PAT innovation around Raman, there are a lot of papers which have been written. you can scroll through these papers, which are there on which are there in Google Scholar, and many of these papers are uh, you can actually download and you can study. And this has been this repository of papers are there for a very long time. So I have just taken some papers here to highlight that how it has evolved. So earlier, Raman spectroscopy was used to study some very basic parameters like glucose and lactate, and people tried to build global models around it, and they try to understand how the processes are working. But now with evolved AI and machine learning tools, we are in a better position to even understand how these parameters are affecting the processes and how we can take control of even the feed and of course the product quality. So let's take this example, which is automated Raman feedback control, which has been the published. It's a published paper by Lonza. So here, the goal one, inline Raman measurement of the critical process parameters, like you have glucose, lactate. I'm sure all biologists talk about it day in and day out. So here, what they have tried to do, what Lonza has tried to do is they tried to come up with very generic models of the key critical parameters, and then they try to put it to multiple CHO lines, multiple CHO cell lines, and try to see how this is actually affecting those CHO lines. So we are in a position to take the feedback control of the processes by just making the models for a very basic cell line. So this is what they've done. The paper is available. You can actually go through it. Now, just to highlight what I've highlighted is the glucose and the phenylalanine. They have studied this, and it very well shows that the generic models worked across multiple CHO lines and MABs also. There is a automated control with Raman, increased productivity by more than 24%, and the critical quality attributes were very well targeted. This is mentioned in the paper. So basically, you develop all these parameters with one or two processes, you are in a position to now extend it to multiple cell lines. So now let's look at this example, which has come from Wuxi Biologics. Again, it's published. So what they have done is inline Raman in CHO cell culture bioreactors. So some of the key basic parameters like glucose, viable cell density, they have studied all this, and they have collected this data using Raman, and then they were pre-trained, and then they were used for the early process development stage. Now, when you are like the previous example, when they are trying to put it for the process development for the other processes, this data that they have acquired really helps in training those models as well. So Raman predicted parameters were fed into an intelligent process development platform. So without any manual intervention, this process was completely implemented in the process. So this helped in understanding specific cell growth, and this also helped in understanding the nutrient feeding pattern required in the process. So all this was happening with just one loop and one pack tool, which basically took the offline values, and the AI and the machine learning tools helped in understanding how the whole process can be controlled, and a proper platform was developed. What this did is that in this process, they have mentioned in the paper that the titer increased by 44% and the specific productivity increased by 42%. And the reducible gains across different clones, because they tried this across different the clone specific data sets, and they found out that the productivity improved in further clones that they worked on. There was 37% titer increase, and this demonstrated applicability across clones with different growth and metabolic profiles. So this they really they explained very well in this paper as to how one data set helped in understanding across different clones, and they were able to do this kind of automation using Raman. Now, this example is from Biogen. So basically, in what was observed here in this in this example is that the legacy process suffered from non-optimized feed strategy. So if something is not optimized, then our nutrients like glucose and glutamine and some of the products like lactate, so they can all, if it is not optimized, and if we see excess of glucose in the process, or excess of lactate coming out, if we don't know what's really happening, then it might lead to the the, the decrease in the cell count, and we can also see the rapid decline in viability. So it's shown pretty well, it's written pretty well in this paper, that how when there was no optimized feed strategy, then excessive glucose led to metabolic inefficiency, and then there was a rapid decline in viability. But when Raman was used here, so automated control of lactate by restricted glucose feeding led to improvements in culture duration, viability, productivity, and robustness. So all these cell culture applications more or less work from anywhere from 15 days to 30 days. So this was extended from 11 to 13 days. Harvest titer increased 85%, and then they were able to understand the feed on demand as to how much feed has to be given, and it also minimized the waste, and it also increased the duration, viability, productivity, and robustness of the process. So this increased the cellular health, stabilized at high levels for the duration. Uh, this is a very good paper where they have explained in detail as to how Raman was used to control different strategies applied in over a period of time, in using different strategies for this process. Now let's look at this application where inline and real-time prediction of recombinant antibody titer. Uh, they, so here the authors have reported that the precision and repeatability offered by Raman meets or exceeds offline methods. This is something very important and very substantial thing to say, which the authors have worked themselves here. The point of saying this is usually your data or is as good as what offline data you give, because Raman will just take spectra. The model is created by correlating with the offline values which are given by the scientists who are working on it. But when they're actually claiming that the precision and repeatability offered by Raman rather exceeds offline methods, it means the predictions are so good with how with the basic model that was developed, that this completely shortens, or rather reduces the need for sampling, and the expectation of the product, uh, and the process that comes out also increases tremendously. So here you can see that batch 1 and batch 2, the concentration, the green line are the predictions, and the dots are the samples that you're taking. As you can see, the predictions are nicely going on with how the predictions should actually look. Now, this paper got released it was written about 2 3 years back, and believe me, it's a very very substantial work. So the paper is written by Daiji Sankyo in Japan. Now, what they have done here is they cultured three different CHO cell clones and in duplicate fed batch cultures in a total of 6 bioreactors, and they have measured whole lot of they've taken whole lot of offline measurements. But in the process, what they have tried to develop is that by taking Raman spectra, working along with the machine learning, they were now able to come up with different strategies to figure out how much feed has to be given, what feed, what nutrients. They were able to learn using the Raman tool here. So Raman predicted surprisingly large number of variables from here. So some of them are the chemical components, like what you can study with their molecular fingerprint, but some of them are secondary tools. But what this algorithm along with PAT tool as Raman could do is that it could surprisingly predict some of the metabolites which they were otherwise not able to detect. So they have specifically mentioned in the paper that the good model was obtained for sodium ions, potassium ions, calcium ions. Some of these inorganic ions they were able to predict, which was otherwise not possible to detect along with so many other components. So they predicted not only amino acid, sugar, cell culture variables, metabolites, nucleic acid metabolites. So they've done extensive work around Raman and machine learning. So they have done some automated model development, because of which the model once developed could be now extended to a whole lot of cell lines. And one very important thing that is mentioned is that it correlates with the bioprocesses that they were working on. But this approach could extend beyond metabolites and predict some of the data and the stress markers which were otherwise very difficult to measure and analyze. So the progression of Raman-based PAT and Biopharma, like I mentioned earlier, it was very basic molecules like glucose and lactate that people used to monitor using Raman, and the integration level used to be single parameter closed loop control. Then with advanced technology, some amino acids along with the nutrients they were able to measure, which helped in multi-feed automatic control. So it's now not just a single closed loop, but we are able to now work on multi-feed automatic control. Then with increased technological advancements, we were also able to study glucose along with amino acids, and this helped in understanding real-time nutrient feeding. But now we are in a position to do some 50 plus analytes along with a whole lot of parameters which are otherwise difficult to measure. And this really forms a foundation for a very comprehensive digital bioprocess control. Real-time release testing has become very much plausible. So some of these publications help us in understanding how Raman technology has evolved in not just to understand the processes, but how to control them, how to have a feedback control mechanism, and how to eventually decide on the process with the full process optimization. And it's also technology to have a faster route to the market. So now, what did we do in all this? So basically, the data analysis, like I said, chemometrics is something which is used in other branches also, but now with AI and machine learning, it has become imperative for probably all scientists to use it. So basically, what we do here is the data which is collected from either chemistry, biology, physics, medicine, whichever field people are working on, the whole huge amount of data that is acquired. So let's say Raman data we are acquiring from some of these processes, then that is correlated with the offline measurements, and the models are built around mathematical tools. Different statistics are applied. So once the models are ready, then we have the predictive models which are connected to the automation through OPC UA server, or different companies have different strategies for it. It goes for automation, and the process decisions are taken. So this is like the summary of what exactly happens behind behind different processes that you're all working on. So the advances in the PAT and future perspectives, basically PAT is a portal to digital manufacturing. So in a nutshell, the Raman probe, which basically collects the data, if we are doing glucose and lactate measurement, this helps in taking the feed control, and it also helps eventually in getting stable critical quality attributes. So any physical process, you have to collect the data, and then you have to train using machine learning. This helps the models which are built, helps in the predictive outputs, and ultimately it helps in the understanding of the process. So basically we are just moving from we the same processes can be replicated. Once the model is ready, we know what is to be taken, how much feed, and what are we expecting from the product. So Raman spectroscopy delivers real value for the life science industry. So it's a because it's a PAT tool, so it's a easier regulatory compliance and consistent product quality improvement coming from the data which we get from Raman. It also helps in faster time to market and greater process efficiency with improved profit margins. So this one of the important things to note here is that it reduces the sampling and sterility risk, because as you know, in bioprocesses, I agree, in chemistry, for example, because of toxic nature of the chemicals, it's difficult to sample. But in bio, it becomes even more difficult, considering um, the huge bioreactors and the cells that you're working on. So there's a lot of sterility risk. So because of the online tools like Raman, you can reduce sampling and sterility risk. It also helps in the scale up and tech transfer, improves in product quality and yield, and it definitely helps in driving the automation. So just to summarize all the benefits that I have just talked about. So Raman spectroscopy is a proven PAT tool with all the benefits which I've just mentioned probably last 20 minutes. Raman helps in situ analytical platform technology to make consistent and efficient and higher quality products. So when we get quick answers, then other testing techniques like wet chemistry or chromatography, because of the nature of in situ analysis, then it really helps in understanding the process or the reactions much faster. Predicting the concentrations, of course, this is what all scientists are looking at, and what we want is accuracy, precision, robustness, and reproducibility of the results. Thank you so much. So you for more information, please, you can contact me, or you can contact my colleagues Shan or Derek, who are working from Singapore.
So, any questions from the audience? You can unmute your mic or you can share your questions in the chat.
Hi, Dr. Shobha. This is Pei Qing. I'm from Cytiva. Two questions on the status of Raman. The
Yeah.
first one is knowing biologics, biopharmaceutical molecules are very complex. I think it's very interesting to hear now there are 50 plus parameters that can be tracked. What is the current status in terms of tracking more complex product quality attributes? For example, aggregation or charge variant for monoclonal
Yeah.
antibody, is that something that can be deconvoluted from the chemometrics? So that's the first one. The second one is, could you comment on the current status of implementing Raman in especially large-scale GMP operations?
Yeah, sure. So yes, so for the charge variants and aggregates, yes, we have. There are some papers which few companies have written. In fact, if I can just take example from a recent paper where they have studied both the charge variants and aggregates, and I agree because this I agree that this is slightly more advanced, and like how initially when Raman technology came into the market and when people started using, they started working on some basic molecules like I mentioned, glucose, lactate. But over a period of time, with more learning, with more experience in the technology, they also move to other parameters. Likewise, aggregates and charge variants, yes, the people have written papers on it and there are companies who are working on it. Um, but I think it will take a little more time for everybody to imbibe this technology. And coming to part number two, there are companies in Europe, US, and few companies in Asia Pacific also where they are using from the lab scale, they move to pilot, and they are now using it in the process scale. So I mean, Biogen, Novartis, they're all implemented it in their process. And I'm sure even Cytiva, if I recall correctly, Cytiva England, they're working on similar process. So one advantage of this technique is this technology is that the whatever data you collect in the lab can actually be used for the process as well to build the model. So that's probably one of the reasons why we have seen Raman being implemented in many companies in their process scale. Did I answer your question?
Yeah, absolutely. Thank you very much.